So this is about blood groups in human beings, and we will discuss about what is ABO system in human beings. This is what I discussed already in my last class, and there are two types of blood groupings done in human beings all over the world, and there are two factors responsible to decide about the blood groupings in humans. One is ABO, other one is RH. So we'll discuss in detail about what are these two types of blood groupings. So depending on the presence or absence of surface antigens on the RBCs, blood group an individual belongs to four different types, namely A, B, AB, and O. So whether a person belongs to A or B or A, B or O group, it is to be observed what types of antigens are present on the surface of RBCs. So, blood group is decided by the surface antigens whichever present on the surface of the RBCs. Likewise, in plasma, antibodies are present. These antibodies are also called as agglutinins. So, please remember surface of RBCs have antigens and plasma has antibodies or agglutinins. So the surface antigens are called agglutinogens also. The surface antigens are also called as agglutinogens, like antibodies which are also otherwise called agglutinins. So these antibodies or agglutinins acting on agglutinogen A is called antibody. So the antibody which acts on antigen or agglutinogen A is called anti-A, and the agglutinin or the antibody acting on antigen or agglutinogen B is called anti-B. And no antigens are present in O blood group. No antigens or agglutinogens are present in O blood group. But O blood group in plasma has both antibodies A and B. Whereas in blood group AB, A and B antigens are present on the surface of RBC, but no antibodies. Neither A nor B is present. So remember, these are the two major differences between the blood groups AB and O. So in O, antigens are absent. Neither A nor B antigen is present on the surface of RBC in O blood group individuals. But plasma of O blood group individuals has both antibodies A and B. Whereas in AB blood group, A and B, it refers to the presence of both A and B antigens present on the surface of RBC. But plasma does not have antibodies A or B. So this is A, B and O blood group system. And A, B and O are actually major allelic genes in ABO systems. We'll discuss in detail in your next class plus 2 about what are these allelic genes of A, B and O. All agglutinogens or all antigens contain sucrose, D-galactose, N-acetyl glucosamine and 11 terminal amino acids. The attachment of the terminal amino acids are dependent on the gene products of A and B. Actually, to produce an antigen A or B, it needs several reactions. Okay, so many reactions, stepwise reactions take place for the formation of or for the synthesis of A or B antigen. So many amino acids which are called terminal amino acids which are required. These amino acids are attached to this galactose or N-acetyl glucosamine. Okay, so it's a briefly it's given the information is very short, but try to understand amino acids or terminal amino acids are required so that they can attach galactose or N-acetyl glucosamine with a precursor substance. This precursor substance is finally will synthesize antigen A or B. Okay, not much information is given. Okay, understand, just to remember as such what is given in your book. So, agglutinogens contain sucrose, D-galactose, N-acetyl glucosamine, and 11 terminal amino acids. So, the attachment of these amino acids are dependent on the gene products A and B. And for these reactions to take place, enzymes are required, and this enzyme for the synthesis of antigens, it is glycosyl transferase. So this is the table to show you uh, what are the types of the blood groups and what are the antigens present on the RBC and what are the agglutinin or antibodies present in plasma. Please go through with my explanation. It will be easy for you. So we'll discuss about this part of the other factor that determines the blood grouping. This is called RH factor. RH factor, it is an antigen, RH antigen or D antigen, which is present on the surface of erythrocytes. 80% of human population, they are RH positive. If this RH antigen or RH, that is D antigen, it is present on the surface of the RBC, such people are called RH positive people. This factor was first discovered by Carl Landsteiner and Alexander Wiener in the blood of rhesus monkey, a type of monkey called Macaca rhesus. Hence, it is named as RH antigen or RH factor. It refers to the antigen called immunogenic D antigen. The individual having D antigen, it is he or she is said to be D positive, RH D positive. If this antigen is absent in the RBC, that is surface of the RBC, then the person is referred as RH D negative. So, rhesus factor in the blood is inherited as a dominant trait. Naturally occurring anti-D antibodies are absent in the plasma of any normal individual. So, this is important information. You have to remember that for antigen D, no antibodies are present in the plasma. For antigen A, antibody B is present in plasma. We learnt already. Likewise, for antigen B present on the surface of the RBC, antibody A is present in plasma. But for this particular antigen, anti-D, no antibodies are present in plasma. However, if a RH negative person is exposed to RH positive blood cells for the first time, then anti-D antibodies will be formed in the blood of that individual. So, you have to remember this. No RH for RH antigen, no anti-D antibodies are present in plasma, but RH negative person, when exposed for the first time to RH positive blood cells, then anti-D antibodies will be produced in the blood of that individual, that is RH negative individual. RH negative individual will produce antibodies for this antigen when exposed to RH positive blood cells. But when RH positive person receives RH negative blood, no effect is seen. Okay.